The title of this talk is Post-Vaccination Infection with SARS-CoV-2. These are my disclosures. So let's discuss cell entry by SARS-CoV-2 just before we move on to the nuts and bolts of vaccination. So of course, SARS-CoV-2 is an RNA virus. It has a 30 kilobase genome. And here we're looking at the spike protein in part A, which is split up into an S1 and S2 subunit. The S1 subunit contains the receptor binding domain, which of course is the target of many of our neutralizing antibodies, as well as an N-terminal domain that is also targeted by neutralizing antibodies. The spike protein is cleaved at the S1, S2 junction, and then again at the S2 prime. The S2 subunit largely mediates the fusion event and has a transmembrane component in it as well. It also has domains that may be involved in immune recognition. So SARS-CoV-2 can infect cells via membrane fusion on the left side in the bottom panel, or by endocytosis, as you can see. And this requires different sets of receptors. For example, TMPRSS2 is critical for plasma membrane fusion and directs infection through that route. Whereas if there is a lack of TMPRSS2 and only ACE2, the main receptor for SARS-CoV-2, then you are more likely to go down the endocytic pathway. So these are all little bits of information that are underpinning the biology of SARS-CoV-2 and also will determine the escape routes that viruses take. On the right here, you can see that there are differences in, for example, the cleavage site, the polybasic region, which is cleaved by furin, cellular furins, and this increases the infectivity of spike proteins. And you can see here that between wild type alpha and delta, there have been changes, particularly at position 681. And of course, in Omicron, we also have a 681H mutation that does likely impact its biology. So here's what's happened to SARS-CoV-2 diversity pre-Omicron. You can see there's a huge amount of diversity with alpha, beta, gamma, and delta here in particular shown, and a huge expansion of diversity. Why do we have this much diversity, therefore threatening our vaccine responses? Well, the answer lies really in chronic infections. It's important to understand that the reason we are having difficulty, let's say, in vaccinating effectively against this virus is that chronic infection with coronavirus is a reality. It's not something that we expected, but in certain immune compromised or subimmune individuals, they end up with a chronic infection over weeks or months in the presence of partial immunity, and this drives virus evolution. On the right here, you can see patients that we studied over time. You can see up to days 101 on the x-axis. And we're looking at spike mutations on the y-axis. You can see that from day 66, we start seeing big shifts in the virus population observed, coincident with convalescent plasma being used in this individual as a proxy for driving immune escape. You can see here that changes at 69 and 70 in the NTD happen, and D796H change. And interestingly enough, these are also mutations that are seen in the Omicron variant. So we observed this more than a year ago in an individual during a chronic infection. You then see further towards the right, you can see big shifts in the virus population occurring with different virus populations replacing the original one. But at the end, when the third dose of convalescent plasma is given to this individual, you can see the original virus emerging, suggesting that the 6970 deletion plus 796 change actually have an advantage in the presence of neutralizing antibodies. And that sets the scene for further variants. This figure just shows you in that individual what happened at whole genome scale rather than just spike. You can see big shifts in virus population over time, groups of mutations coming up and then down. And this is suggestive of haplotype competition within the host. So the new variants are arising as a result of mutation and selection, and then a sort of evolutionary struggle between different virus species within the individual. And eventually the fitter one will probably transmit and then set up a chain of transmissions that lead to the new variant. So this process originally led to a number of variants of concern, and these were characterized by the N501Y mutation, alpha, beta, and gamma. Here you can see with a number of changes in the spike protein, and early data from a number of labs, including ours, showed that the alpha variant, for example, which had the N501Y mutation, along with the delta 6970 deletion in the NTD, as well as a number of other spike changes, was actually fairly sensitive to neutralizing antibodies that were elicited either from previous infection. Here you can see in convalescent sera, and also following vaccination with mRNA or protein-based vaccines, we got a fold change of only around twofold loss of sensitivity of alpha. And this was relatively reassuring. But of course, we then had to face the beta and the gamma variants, and this figure shows you some data comparing alpha, beta, and gamma. Here we're using the old Pango nomenclature for those variants, B117 for alpha, P1 for the gamma variant, and B1.351 for the beta variant. You can see the beta variant there in red is the least sensitive or most resistant to neutralizing antibodies directed at the receptor binding domain. And of course, data from our group later then showed that age has quite a significant impact on neutralizing responses. Here, if you look at the first dose to response to Pfizer vaccine after the age of 80, you get quite a precipitous drop. This is partially rescued by the second dose, but of course, we envisage that the decay of immunity will probably be faster in that group. And this figure really is designed to also show you that the advent of variants has increased the susceptibility of the elderly age group to SARS-CoV-2. In other words, the neutralizing antibodies are less effective because their titers are generally lower, and therefore variants pose a significant threat to those with suboptimal immune responses. What about real-world efficacy of the vaccines? Well, we can see, of course, here that the B117 was highly preventable through vaccination, with data here for Pfizer, Moderna, and the AstraZeneca vaccines showing very good protection levels. Against the beta variant B1.351, of course, there was a bigger problem against protection for symptomatic infection. Of course, protection against severe disease was excellent across most vaccine platforms. This leads us to some nice synthesis of data from a couple of groups, where they've shown here on the left a paper comparing in vitro neutralization on the y-axis, in other words, predicted efficacy against observed efficacy in clinical studies. And you can see that most of these vaccine platforms fit somewhere along this line, suggesting that there is a correlation between neutralizing antibody responses elicited after vaccination and real-world efficacy, in other words, protection against symptomatic infection. This is not severe disease, remember. On the right here is a particular piece of data from the ChAdOx1 studies where they prospectively managed to identify a titer at which the risk of infection potentially increased significantly, and that's a titer of around 185, or a serial dilution of 185. So we can actually put numbers on the protection threshold at which the loss of protection becomes quite significant. With the advent of the Delta variant, which arose in India after a competition between 3 different sublineages of B1.617, the 617.2 emerged as the winner, as it were, and disseminated not only throughout India, but of course the rest of the world, as we know. In the right-hand panel, you can see the shift from orange to green viruses, and those represent the B1.17 variant predominantly before, and then, of course, after we have the Delta variant in green. So what about the Delta variant responses to neutralizing antibodies? Well, you can see here that using live or pseudotype virus, there is a loss of neutralizing sensitivity of Delta. It's quite modest. It's certainly not greater than 10, which is what we were seeing for the beta variant, but it is nonetheless significant. And we believe that part of the reason Delta was able to become so successful in previously infected and even vaccinated individuals is that it has very, very high infectivity. Data here is showing that on the left, the virus 617.2 has much higher levels of replication compared to the alpha variant B117 on the left-hand side, and also in human epithelial cells, which is a more physiological model for airways. You can see again that the 617.2, in other words, in green, those bars are much higher than the orange bars and on a log scale, suggesting that the Delta variant has a significant replication advantage. We can use more sophisticated models, such as airway organoids that are derived from stem cells, to show pretty much the same thing, where the 617.2 or Delta variant is far superior in replication, both within cells and in terms of secreted virions in culture supernatants in this model system. This is assisted, let's say, by the phenomenon of syncytia formation, which I believe aids immune evasion of the Delta variant compared to wild type. You can see here a system we have for lighting cells green when they fuse together, and we can show here that the B617.2 or the Delta variant is far superior as compared to the original Wuhan 1 virus when it comes to fusing cells together. The y-axis shows the green fluorescent protein, which is a marker of fusion, and you can see over time the B617.1 is able to achieve higher rates of fusion and reaches higher overall levels of cell-to-cell fusion. And if you imagine, this is a method potentially for the virus to move from one cell to another to spread an infection without having to leave a cell, and therefore does not have to encounter neutralizing antibodies that are elicited from vaccines. So all of that in vitro data put together nicely explains what we observed, which was really for the first time high levels of vaccine breakthrough. This is an example. These are the first data published in the world on vaccine breakthrough with Delta, and they came from India, where Delta obviously emerged. And in healthcare workers, you can see infected with different virus lineages. You can see this epidemic sort of curve on the left-hand side with a mix of lineages. But if you look now on the phylogenetic tree on the right, you can see that in those that are double vaccinated, in other words, dark blue, you can see the number of days elapsed since the second vaccine. It's around 40 or 50 days, which is a good time to achieve peak neutralizing antibodies. You can see that there's a large number of infections with Delta, almost identical sequences indicative of a super spreader event. In other words, many people who are double vaccinated being infected with relatively high viral loads despite being vaccinated in a hospital setting. So rather worrying data, which then were seen throughout the world in populations. One silver lining to this is the fact that although people start off with very similar viral loads early on, as you can see, the red dots and green dots are virtually overlapping. Those that have vaccine breakthrough, in other words, those who are vaccinated, experience a faster decline of the amount of virus in nose and throat swabs as compared to those who are unvaccinated, who are shedding for longer periods as compared to their vaccinated counterparts. So this potentially will have implications for transmission. Importantly, one must note, though, that up to day 4, the viral loads are pretty much the same, and those are the critical few days when people are asymptomatic and could be transmitting unknowingly. On the right are some data looking at how probable it is to culture virus in the context of breakthrough in vaccinated versus unvaccinated individuals. And you can see here that the chances of isolating virus in culture is lower if the virus has come from a vaccinated individual, which makes sense in that the virus decays faster, it may be coated in antibodies and less viable for infection. So that's good news in terms of vaccination. These are some data looking at whether we can actually assign a probability that one vaccinated person has transmitted to another individual. So that's obviously one of the key insights that we need to understand. Are those people with high viral loads actually infectious for other individuals? And we derived this information from the healthcare worker study I showed you earlier. And what we've done here is formed a network analysis with simulations to understand the probability of transmission based on genetic distance between sequences in infected individuals, the date of infection, the serial interval, and parameters identifying how likely it is that there was a transmission. So you can see here in red, these are individuals who have been doubly vaccinated more than 40, and the last dose more than 14 days ago. And you can see that there are thick black arrows emanating from at least 2 of those individuals, 205 and 207, suggesting that these are very likely to have transmitted to another individual, lending support to the fact that these high viral loads are in fact associated with transmissible virus. So what about vaccine waning? Well, you can see here that, of course, over time, this is an analysis that was in the Financial Times, but actually the source was University of Oxford and the ONS, where they've shown that as time goes on after the second dose, you can see that the efficacy against symptomatic Delta infection falls rather gradually for AstraZeneca, but more steeply for the Pfizer-BioNTech mRNA platform. And you can see that this, by 3 months, the protective efficacy against symptomatic infection has dropped quite significantly. These are data supporting those UK data, and these are now from the US and a much larger or a very large cohort of individuals in the Kaiser Permanente healthcare system. And you can see here they've compared the Delta variant efficacy as time goes on after the second dose, and they've also looked at other variants in red. The adjusted vaccine effectiveness is plotted on the y-axis, and you can see that Delta lowers the overall efficacy of the vaccination, as we would expect from the data represented, but both Delta and non-Delta show similar declines or slopes over time since second dose. So what about severe infection? I mean, you can see, you can argue that symptomatic infection wouldn't be that much of a problem. Of course, you have the issue of transmission being more likely where the neutralizing titers have fallen. But here are data from Israel showing that in fact the number of severe cases started rising in vaccinated individuals, and it was seen predominantly in the older age groups. And of course, Israel was one of the first places to use the mRNA BioNTech vaccine early in the year, and therefore they had experienced waning before many other countries. These data show what happened after the third dose was administered to those individuals, and you can see that there's a very marked reduction in the rate of severe deaths or severe illnesses and deaths in this case in the over 60 age group after the third dose. You can put some numbers on that. This is data from the NEJ and Bar on paper showing you that the booster reduces the rate of severe disease both in the 60 plus and 40 to 60 age groups, with quite significant rate ratios here of 18.7 and 22 respectively for the 60 plus and 40 to 59 age groups. So boosting appears very important, not only for reducing infection, but also symptomatic and severe disease. So now bringing this together, we can now see that pan-coronavirus vaccines appear to be desirable because if you're getting a loss of protective efficacy as we see more and more variants. We've seen Delta putting a big dent in the vaccine effectiveness of our current vaccines. We now have Omicron, which is predicted to have less efficacy, or to be less sensitive against neutralizing antibodies and potentially T cells. So we do need to get towards pan-coronavirus vaccines. This is one example where the group have targeted a specific epitope or area within the receptor binding domain, spike protein that's shaded in blue here, which means that it shows very little variation across global sequences, suggesting that it's highly conserved, and therefore that mutating any of those incurs a significant fitness cost for this virus. And therefore these immunogens may be the optimal future targets for vaccines. Okay, now we of course need to acknowledge that we have Omicron B.1.1529 with a large number of new mutations spread right across the virus in some key positions, not only affecting neutralizing antibodies, but likely affecting cleavage and maturation of the virus. And this virus likely has immune escape properties, but it's less clear as to how infectious this virus is. So to wrap up, I've shown you that the alpha variants showed modestly reduced susceptibility to vaccine-elicited antibodies, and as variants have gone on, we've seen greater and greater losses of neutralizing sensitivity, and this really is a signal that we need to develop pan-coronavirus vaccines in order to limit virus escape and to maintain control of the COVID-19 pandemic. Thank you for your attention.